revolves around our life, which is the central dogma of molecular biology. Why it is known as the central dogma? It is known as the central dogma because around this dogma the whole life revolves, which starts from a DNA and ends on a protein. This is known as the central dogma because it involves the formation of a protein. Protein is the expression of a body. That means if a right kind of a protein is produced by a body is said to be a healthy body. And if a wrong protein is formed by a body, a defaulted protein is formed by a body, then the body is said to be diseased. So this is formation of a protein from the basic unit of a life, that is a DNA. So the central dogma of molecular biology involves three processes, which is one is replication, second is transcription, and third is translation. The replication involves the formation of a DNA from a DNA. Then second process, transcription, involves the formation of an RNA from the DNA. And this RNA acts as a raw material for the formation of a protein in the process of translation. Now let's understand these processes one by one. The first important process which is involved in the central dogma is the replication. In this replication, the double-stranded DNA is converted to another double-stranded DNA. So a DNA to a DNA. This is known as replication. There are certain prerequisites of a DNA replication. Of course, yes. If a replication is occurring, any process in the body is occurring, it will involve certain energy, certain enzymes, and a primer template. Of course, yes, a template, without a template, no process can start. So, the prerequisites of DNA replication are, first is 4-deoxynucleoside triphosphates, which is GTP, CTP, ATP, and TTP, which are the base pairs, nucleotides, which are involved in the process of chain building. Okay, the chain is formed, but how? Of course, yes, a junction is required. That junction is known as a primer template junction. Okay, primer template junction is there, nucleotides are there, but how will the process initiate? So the initiator proteins are involved in the process of replication. Now let's understand what is a primer template junction. A primer template junction is actually the junction between the double-stranded DNA and the single-stranded DNA. Okay, at this junction, DNA polymerase comes and binds and starts adding nucleotides. Once it starts adding nucleotides, then the phosphodiester bond needs to be formed. Of course, yes. Otherwise, how the nucleotide chains will bond and how that another strand of DNA will form and how the single stranded DNA will convert into a double stranded DNA. So for that, magnesium ions and zinc ions are required to form the phosphodiester bonds. And certain initiator proteins are required, of course, yes. So these initiator proteins are divided according to the process of DNA replication. So first process of DNA replication is that a double-stranded DNA, the template DNA, the mother DNA, parent DNA is converted into a single-stranded DNA. So first the DNA helix needs to be unwounded, and this is actually achieved using helicases enzymes, which is a hexameric protein and it uses ATP. While in prokaryotes, these helicases are known as DNA A and DNA B. Now, of course, if the helix is unwounded, if any spring, for example, is unwounded, then of course a supercoil is formed in that case. So that supercoil needs to be removed. Similarly, when a helix DNA is unwounded, the supercoils are formed naturally. So these are unwounded by the use of topoisomerases in the case of eukaryotes and chiarases in the case of prokaryotes. Next is the prevention from recoiling. The supercoils have been removed, but now this DNA can recoil anytime because it has a natural tendency to stay in a helix form. So it is removed, it is prohibited by the use of SSPs, which are single-stranded binding proteins, which stick to the single strand so that they may not rebound. Next is the formation of a primer. Of course, if a primer is not there on the template, then how will the replication process carry forward? So this is formed in prokaryotes by the formation of binding of primase enzyme with helicase enzyme, forming a holoenzyme which is a primosome. And in the case of eukaryotes, this primase is intrinsic to polymerase alpha, and this polymerase alpha, when bound, forms the primer template junction in the case of eukaryotes. There are many types of DNA polymerases in the case of eukaryotes and prokaryotes, which actually moves on the single-stranded DNA and forms another strand and replicates the DNA. So in eukaryotes it's of 6 types, and prokaryotes it's of 5 types. In eukaryotes it's the 6 types which forms the holoenzyme are polymerase alpha, delta, epsilon, beta, theta and gamma, while in prokaryotes it's of 5 types, which is 1, 2, 3, 4 and 5. Now we have explained everything. Okay, now the DNA polymerase as it moves, then it forms a replication fork. Why it forms a replication fork? Because when it will move, when the double-stranded DNA is converted into a single-stranded DNA, then it will be 2 chains. One is 5' to 3' and one is 3' to 5'. So once it will move, it has a natural tendency of replicating the DNA from 5' to 3'. But if it is carry forwarding the replication, of course the 3' to 5' chain will also replicate because nucleotides are added. So that chain breaks in between and forms the fragments, which is known as lagging strand or Okazaki fragment. And the normal strand from 5' to 3' is known as a leading strand. So the direction of replication is both from 5' to 3' and 3' to 5', forming the leading strand as well as the lagging strand. You need to actually understand how the primers are formed and how the primers is removed after Okazaki fragment is synthesized. We will study it in the termination process. So this is the overview of a DNA replication. You can see the formation of an Okazaki fragment. You can see the DNA polymerase is there. Okay, you can see it once again. Now once this is formed, we can go to transcription. In transcription, the DNA which is formed is converted into an RNA, so that RNA can be used as a raw material in the process of translation. It is actually very less accurate than replication. Now, since the DNA was replicated by DNA polymerases, RNA is replicated by the use of RNA polymerases. This RNA polymerase is a holoenzyme which consists of 5 subunits: alpha, alpha 2, beta, beta dash and omega. Alpha 2 and alpha are the 2 alpha subunits which bind to regulatory factors. This subunit is not used on promoters without an up element. The beta subunit has the polymerase activity, which is actually involved in the conversion of DNA to RNA. It catalyzes the synthesis of RNA, which is the main unit. Beta dash helps in the binding of RNA polymerases to the DNA template, while the omega restores the denatured RNA polymerases to its functional form in vitro, which can be used again for another process of transcription. Let's understand the process of transcription separately in eukaryotes and prokaryotes. There are many types of RNA polymerases in eukaryotes. For example, RNA polymerase 1 synthesizes pre-rRNA 45S, which matures into 28S, 18S and 5.8S rRNA. RNA polymerase 2 synthesizes precursors of mRNA and more single nucleotide RNA and microRNAs. RNA polymerase 3 synthesizes tRNA, rRNA, 5 subunit and other small RNAs which are found in nucleus and cytosol. Then there are two RNAs which are found in plants, which are RNA polymerase 4 and RNA polymerase 5. Now we'll move to steps of transcription. The basic three steps of transcription are initiation, elongation and termination. In elongation, this process, which is actually demonstrating the prokaryotic initiation and elongation, which involves the formation of an open complex, which converts into a closed complex, and the cycle continues. So first, a closed complex is there, which is formed during the replication process, converts into an open complex, which carries forward the initiation process of transcription, then the elongation, termination, again a closed complex. So a recycled chain occurs. In eukaryotic transcription, the transcription requires certain factors, which is a Tata box, which is the closed complex, enhancers, energy in the form of ATP, and certain transcription factors, which are TBP and transcription factor 2D, 2A, 2B, 2F, 2E, 2H and 2J. This is the whole process which involves the formation of the RNA. This is the elongation and the initiation process, which involves the Tata box to initiate the process of transcription. Now, once this transcription is there, you must be wondering how you have to complete the whole syllabus in just a small time, because time is like 6 months, 5 months is very less to read such huge topics. So there are certain facts of GATE, BT, GATE Excel and CSIR examination which is being designed by our faculties at CTech SPS, so that the students can even concentrate on these subjects. They can keep these subjects on tips and they can clear the GATE examination and CSIR examination easily. So these are the facts of GATE, BT and GATE Excel examination. And these are the facts of CSIR Life Sciences examination, which you can easily get on our website www.ctech-sps.org. Now let's come back to the topic, which is the process of translation. We have already studied the process of replication, where DNA was formed from another DNA. Then the process of transcription, from which the mRNA was formed from the DNA. Now this RNA will be utilized, which is known as mRNA. It will be utilized to form the protein, the basic essential unit of life. You can see the basic outline of translation. This is the subunits, ribosomal subunits, which are actually involved during the process of translation in eukaryotes as well as prokaryotes. You can see how this 70 subunit in the case of prokaryote is divided into 50 S subunit and 30 S subunit, which are separate subunits. Separately, in the case of eukaryotes also, 80S ribosome is there, which is divided into 60S subunit and 40S subunit, which has separate smaller subunits. Altogether, the smaller subunits and the larger subunit bind together to initiate the process of translation. Now let's understand one by one the processes of translation. First is the initiation process, which involves the protein synthesis in eukaryotes. It involves the interaction between a poly-A chain and the initiation complexes, which are actually required for the stability of mRNA, because mRNA is not at all stable. So it needs to be stabilized first, then only the protein synthesis can carry on. And in eukaryotes, it carries on an endoplasmic reticulum. Then second is the elongation in eukaryotes, which you can see involves many of the elongation factors, which are factor 2, 3, 4b, 4a, 4e and 4g, which forms the holoenzyme. This is the complete process of translation in prokaryotes, which involves the most important thing, which is the initiation codon, which is AUG. The main difference between the translation in prokaryotes and translation of eukaryotes is that the translation in prokaryotes involves F-met RNA, while the translation of eukaryotes involves only methionine-induced RNA, met RNA. The translation in prokaryotes involves the Shine-Dalgano sequences. This is the termination process, which involves the stop codon. Okay, the start codon is there, in which the mRNA can translate into the protein chain. But once it is translating, the RNA polymerase is coming, transcription is going on, then the translation is going on by the elongation factors. Once these factors are continuously converting this RNA chain into a protein, then if it has to stop, a proper protein required for the body is formed, then stop codons comes. So the factors are not able to translate them because they are not able to read these codons. So because of that, these are known as stop codons, which are of three types: UAG, UAA or UGA. Once it is stopped, release factors comes. Release factors, it releases all the subunits, the mRNA template and the free polypeptide nascent chain. Now since this chain is nascent, it needs to be undergo post-translation modifications, so that it is very nascent, it doesn't know anything, it is in the word now, so it has to be prepared for the body. So you can see this, the nascent protein is there, it undergoes protein processing, converts into a mature protein, which folds and form a biologically active protein, which are certain hormones in the body, enzymes in the body, which are used in a daily process by the body to conduct daily activities. These are the few things which are important for a protein. Stability of a protein, biochemical activity should be normal, protein localization should be there, where it has to go and act, and so this protein signaling. It has to interact with other proteins, interact with the body, form a cascade of enzymes, form a cascade of activities. Now next is the protein turnover, which is a protein degradation. Involves ubiquitination and spontaneous oxidation. This is the basic process of ubiquitination, which involves the activation and the attachment of ubiquitin to a protein. Another is The other processes are hydrogenation and sulfation or methylation. Once the protein is not used by the body, unused protein is undergone protein degradation, otherwise it will start causing the disease. So these are certain factors and the processes which are involved in the degradation of a protein. Thank you so much, students, for going through this PowerPoint presentation. I hope it will be very useful and beneficial for you to read the subject, the whole topics of transcription, translation and replication. For more PPTs, you can log on to our website www.ctech-sps.org. Students, the CSIR examination is coming up for June 2013. So you can start your preparations from today itself full-fledgedly, so that at the last moment there is no hurry-burry. You can only prepare the question papers at the last moment rather than completing the syllabus. So you can join the classes which are conducted by CTech SBS. The classroom coaching program is starting from the 10th of February 2013.